exam is this Friday.
What?
Oh, yes, sure. How will it work? You will get, there will be 22 questions, there are 8 pages. 15 of the questions are multiple choice, and that's worth half of the points of the exam. There are then 7 open questions. Some of which are short answer, where you're supposed to keep the answer really short. And some of the questions are what I call problems and diagrams, where you have to make a drawing or part of a drawing or fill in a drawing and give a little bit of explanation as well. So, we give the exams back to you in the labs, and in order to help us organize that process, we collect the exams, the written part, grouped by the lab number. So we will have 8 stacks here for you. And if you're in lab number 1, you put your exam in stack number 1. Now, you also have a multiple choice answer sheet, and that answer sheet goes on a separate stack. So you turn in the answer sheet separately from the written exam. Okay? It's very important that you put your name on the exam as well as your student number, and you have to do that on the answer sheet as well as on the written part that you turn in. So both the multiple choice and the regular exam questions need your name and student number. There will be 2 versions for the exam, A and B. You also have to put that on the multiple choice answer sheet. So, I don't think there's especially a reason to freak out. It should be doable. I know it was last year. The exam is about the same length. I have posted a practice test on the web, and it gives a pretty good idea of the format that you can expect. Do not expect to finish the exam in 30 minutes, even though I'm sure some students don't do that. I think you might take 40 to 45 minutes. And some of you might take close to 50, but I think it's doable. I think it's doable. Okay. Now, okay. Today we'll do a little bit of a review of some of the material that is on the exam. Tomorrow at 5 o'clock I will also be available in room Physiology 103 from 5 to 5:50 to answer any questions you might have. And I can then answer them to you as a group, or you can show up at office hour tomorrow afternoon or Friday morning, I think. All right. So let's do some reviewing. And important in section 1 was how cells are studied. And part of that is to look at cells and parts of cells. And so we can look at some examples of microscopy, and we can always ask ourselves the question, what type of microscope might have been used here, and what was the contrasting mode that was used? I understand that this is a stupid question in real life, because in real life you would have made this picture yourself, you would have known which microscope you went to. But just for us, it's a little bit different, so let's just ask ourselves that question. So here we see a filamentous algae. It's a eukaryotic cell. It has here chloroplasts, or maybe a big chloroplast. And what type of microscope was used here? What would it be? So, is this an EM picture? Does it look like an EM picture? No, it's not. And also, we don't see a scale here, but really, this is not very high magnification, it's maybe 50 to 100 times. I'm going to put you in the slide guard. Enjoy the pictures. So this can be a light microscope. And also the image is transparent, which tells us it's a light microscope. This object has pigments itself. This is just something we can view with regular bright field. Alright, now here we have a set of cells, HeLa cells, and you can see here the nuclei in red, and we can see different cytoskeletal elements that are stained with blue or with yellow. And so what type of microscope would this be here? This is a fluorescence microscope. Another question is, do you think these cells are alive at the point that the picture was taken? And if you think yes or you think no, then think why. So what are we saying here? Who says that this cell is maybe alive? And who thinks it's probably not? Honestly, it's difficult to say, but if we know a little bit more about the method, so we could have used some fluorescent gene fusion system to stain these cytoskeletal elements. So if this was tubulin fused with GFP, we could have seen it in a live cell. But the nuclei, to get them stained like this, we probably added some kind of a dye, a fluorescent dye. And so I think these cells are killed. There was a staining molecule added to the cell, so they are probably not alive. So they're fixed, and probably the cytoskeletal elements were stained with the help of an antibody, a fluorescent antibody. So these cells are then not alive. If I would give you the detail that these cells that the structures in the cell were visualized using antibodies that have fluorescent groups attached, then you know that the cells must have been fixed and therefore they're not alive. Okay, so that's that one. Now, this is an image that was taken 1 or 2 years ago in the cell biology labs. And it is a fluorescence microscope image. It was used with the fluorescence microscope in the lab. Here you can see yeast cells. And you see there's a GFP expressed in the mitochondria. See those little mitochondria in this cell? There's a cell that's budding. So we can see fluorescence in the lab. This is what fluorescence looks like. It's a fairly weak signal. That's often the case with fluorescence. That's another strength of using a confocal microscope. The confocal microscope just scans this image several times, scans this specimen several times. If it doesn't, this is a regular fluorescence microscope. If it were confocal, by scanning over and over, you get a good amplification of the signal where there is one. So you increase the contrast with that. What you can also notice, there's all this stuff here in the background, and this, I'm suspecting, is actually, some of these might be yeast cells that are just out of the plane of focus. So we're having this limited depth of field here, because it is still a light microscope. Okay, this is a picture of research in my own group, actually, where we've taken an isolated plant cell, and this is a bright field image of that cell. We've removed the cell wall and made a bright field image, and this cell was actually transformed, transfected. We added DNA to it, the cell takes it up, and it then expresses proteins encoded by the DNA that we give it. And that protein was fused to the green fluorescent protein, and we see that green fluorescent protein image here. So this is a confocal microscope image. And the way this image was formed is that the confocal microscope was scanning at different levels of depth in the image. So you get stuff here in the background, there's something here. This is a full sphere, this cell. And in the bright field, you just see a circle, but you know it is a sphere. So we have here a structure, it's actually a chloroplast. It's in the background. And we get this in nice focus because the confocal microscope first scanned at this distance away from the objective lens, and then it scanned closer to the objective lens. So at different depth situations we have scanned this, and then everything was overlaid. And that's why you get this very, I think, crisp image. This is the fluorescence from chlorophyll. If we have chlorophyll, which is absorbing in the blue and in the red, if we shine blue light on it to excite, it fluoresces red. So that red fluorescence is chlorophyll. And here we have the overlay of the red fluorescence from the chlorophyll and the green fluorescence from our protein. So we see that we get the green fluorescence very close to the red fluorescence, which means that our protein, in this case, is probably in the chloroplast, but not in the thylakoids itself. The chlorophyll is in the thylakoids. Within the chloroplast, our protein is probably in the stroma. Does that make sense? To give you an idea of how that image is formed. So here we have another image, and here we see again microtubules, and I think they've been stained with an antibody. So how was this image made, and is this cell alive? Let's take a vote on, is this cell alive? Let's say yes or no. So who's yes? And who's saying no? This cell is clearly dead because it was stained with an antibody. So we had to make holes in the membrane to let the antibody in. And so that antibody must have had a fluorescent group attached. So this is a fluorescence image, and probably a confocal image because everything is very crisp. There's really a lot of detail that we can see, and we don't get a lot of blurriness if we think about the depth of the cell. And yeah, and you can see here we're at 15 micrometers here for our scale bar. And so yeah, the resolution of any light microscope is 0.2 micrometers. So we're well above that resolution. So this is a light microscope that must have been used here, or that could have been used. Now here, if we look at this situation, we have, there's a yeast cell, and there we have a white blood cell, a type of macrophage that is actually ingesting first this yeast cell. And if we look at the scale bar here, we see that we have detail in the image that is far below 1/10 of this scale bar. This can never be a light microscope. So this has to be some form of EM. And in this case, it's a scanning EM image. I know a lot of students ask me the question, how can I sometimes see the difference between a scanning EM image and a transmission EM image that is taken after fracturing and then shadowing? Because both have a three-dimensional field. And then I can only think of this answer. The scanning EM pictures really look like we're having an object in three-dimensional space that kind of sits in a box, that sits in a room. This is clearly a three-dimensional object that sits on top of a surface. And in the case of fracturing images, we can imagine that we're looking down on a mountainous landscape from an airplane. And I think that is the only way I can tell you how you would be able to tell the difference. And of course, in real life, a researcher knows which microscope he or she is walking to. So this has to be a scanning EM image. Now, where is the detector? So remember, the primary beam of electrons is coming down from your point of view onto the object. And those primary electrons collide with electrons that are present in the specimen. These cells were fixed and they were covered with some form of a heavy metal. Often that's platinum. So those heavy metals have a lot of electrons that go around their nucleus, and there's a good opportunity for the primary electrons to bump into them, and that leads to the emission of secondary electrons from this specimen. The secondary electrons then reach a detector, and that detector is just a positively charged electrode. And when the electrons hit it, you get a current, and that current is translated by a computer into a signal corresponding to brightness. So when we're having the primary beam focused on one point, like here, we're not putting out any secondary electrons, so that the image there remains dark. But when we are actually, we might be putting out secondary electrons, but they're not captured by the detector. If we're over here, apparently, we're putting out a lot of secondary electrons that get captured by the detector. So the detector has to be on the top. And somewhere on the top is where the detector seems to be. So if you understand that, you understand how the image is formed. So scanning EM pictures look like three-dimensional objects that are in a room or in a box where there's illumination from the point where the detector is. So scanning EM image. Interesting here is, you know, yeast, it's budding yeast. And this is a yeast cell that has budded before. We see here the bud scars where daughter cells have actually been pinched off. And that causes some damage in the cell wall that's then repaired later by the deposition of chitin, and you can see that in scanning EM. We call this the bud scar. Alright. So here we have a virus body. And we see here the scale bar, it's 0.1 micrometer, that's already by itself smaller than the resolution of a light microscope. So this can only be in EM. So in the EM we have thin sections, we have negative staining, and we have shadowing. And this looks like a clear thin section. So we were pretty lucky, or the person taking this picture was pretty lucky in hitting this cell where we have viruses budding off from the cell. You see here, this is one cell, and this is the plasma membrane. So really capturing a virus here in the act. And you can actually see the virus is approximately 0.1 micrometer in diameter, right? And we're actually, apparently in this section, have captured part of that curvature of that virus. So we actually see some metal attaching here to this virus. We see all the spikes going all around. So lucky shot though. So we can capture all these viruses in this very thin section. That's probably thinner than this 0.1 micrometer. Okay, this is then another image of a bacterial virus that's coming out of a bacterium. So here we have our bacterium, and this is an E. coli, so it has an inner membrane and an outer membrane. That's just what E. coli bacteria have. We're seeing here that the phages are being assembled, and these are the old phages. And these phages have infected this cell, they have injected their DNA. That DNA has replicated in the bacterium, has been transcribed and then translated. And that translation leads to the formation of phage heads which are being assembled. Ultimately, this bacterium will fall apart, will lyse, and that's how the phage particles, so the bacterial viruses, get out. So this is, again, this has to be a thin section. And again, it's a pretty lucky shot because we see here a couple of these phages actually sitting on the surface of this bacterium. Okay, there we have our same friend, the virus, the bacteriophage, but here it's just in isolation. So it has to be an EM picture. But is this then a scanning EM or a transmission EM? It clearly looks like a scanning EM. We feel that we have this three-dimensional image in a box. Open it up. Here we have once again the same bacteriophage, and here it's mistreated a little bit with some chemicals, and it spilled all its DNA out. So all of this DNA here, it was all in this phage head. Can you imagine that? So this has to be an EM image. And what is the method of contrast here, you think? Has to be negative stain. Yeah. So I'm not sure if I can get this whiteboard to work. I was giving you the shot before, and it didn't work for me. You don't see that, right? I need to use the cursor. Okay. So I can try to explain how that negative staining works. So let's just say we've isolated that phage. And what we're doing is we're putting that phage on a thin surface, which is typically we use carbon, very thin layer of carbon film. We use carbon because it is low atomic mass, so it's transparent in the EM. So on the top of that surface, we put down our isolated bacteriophage. I'm trying to just put it down there, okay? That's our phage. It's going to be challenging to draw it. So remember that in the EM, this bacteriophage is pretty much transparent. It's invisible because all the atoms are of low atomic mass. Because we have DNA and protein there. So we have the elements hydrogen, carbon, oxygen, nitrogen, and phosphorus. So that's not visible in the EM. So what we have to do is we have to cover this with a metal salt. So if I just pick the color green here, and say, okay, we deposit metal salt particles. We first have a solution, and I'll just throw in a couple of metal particles that are in solution. Then we let the solution dry and the water evaporate, and what will happen is we'll form a layer of stuff that's just pretty much everywhere, and then it runs up to our specimen. I'm just trying to fill that up. Oh, you have to be patient with me. And the reason is that this board is not working. And I've called classroom services about it. And right now they didn't fix it. So anyway, we deposit our metal there. That's what happens after things dry out. So now if we put this in the EM, we will have our primary electrons that run through specimens. So we're going to take our primary electrons here, and in red here, there comes a primary electron. And it is stopped right here, so this primary electron will be scattered away. It will stop. But if we have a primary electron that runs right here, it will go right through the specimen because we're of low atomic mass right there. We don't have the metal deposited in this area. So wherever we have organic material, we get a brighter area, and wherever we have our shadow, our contrasting metal deposited, we get a darker spot. So it will be dark here, and it will be bright over there. And then whatever electrons make it through the specimen, that's just captured by the objective lens, that's magnified, and you see that on the screen. So that's the idea of how the method works. Okay, so we have here, where are we? Over here. Come on, do this. I'm not sure what I did. Maybe I have to come. Technology can let you down, you know? Okay, that's better. I don't know. It's kind of stuck there. So I hope you can still see this. Can you? Okay, so we have here, once again, you see the DNA there, that's white in the darker background, darker grainy background. You see the same for the phage. So if we go here, we have a thin section again of my friend, the tight junction. Remember the structure of the tight junction? And we can look at this structure also with another method using fracturing, freeze fracturing. So here's the freeze fracture picture. I'm going to try something. Let's see if I can get this back up. So how is this image formed? We have a couple of cells that are frozen. We hit the frozen object with a very small hammer. Water is in contact with water by means of hydrogen bonds. These are pretty strong connections, and it's hard, therefore, to break through the ice. Actually, you all know that, but if you have an ice cube, that's pretty hard to break that. But the two leaflets of the biological membrane are not strongly connected. So we get preferential breakage right through the central plane of the membrane. And so we're breaking through the membranes. So we have here a cell that's on top and a cell that's at the bottom. We're here in the membrane of the cell on top and the membrane of the cell below. Here we run into the tight junction. Here we're still in the cell on top and we're going there and over into the cell below. And these little ridges, that is material that sticks out from the lower cell. These are the proteins that form the tight junction. So fracturing doesn't like to happen through proteins. You always fracture around the proteins, so the proteins tend to stand out. And we know that the shadowing source here has to be on what is the left side. And between an image like here and an image there, we can form a three-dimensional image of what the tight junction looks like. And the function of the tight junction is to make sure that in an epithelial tissue, that all the transport has to happen through the cell layer. Yes, you had a question.
Could you just explain a little more what the shadowing source consists of?
The shadowing source is, in the case of fracturing, it is a metal. It's often platinum or silver. Platinum is often used as metallic platinum, which is just heated up, and then individual atoms just jump out of that metal. And they just sort of rain down under an angle. If I would draw that on the board, which I think is a good idea in these circumstances. So light come on. So if we have our object here, and maybe we have some breaking surface, we have actually within an area where you keep a very low pressure, so vacuum, and you have a cooling mechanism, so this biological sample remains very cold, so it's not evaporating away. You have here a little bit of metal, and it's just being heated up by means of a set of electrical wires. And individual atoms just sputter off, they just come off, they go in any direction, but for the specimen that means that they're coming down under an angle. So we get accumulation of staining material wherever we can directly hit the surface. So we get very little accumulation here. So we get an area that in the EM later becomes dark, and other areas where the electrons come through, so it becomes more light. So actually these electrons didn't go through. Stop. Make sense? Okay. So here we have another virus, and this one is also negatively stained. Yes?
Back to the shadowing image. So if the darker side is the side from which the source came, back to the freeze fracture picture, wouldn't it be coming from the right side?
If we look right here. So from your perspective, the way to look at this is, if you look at a very small particle somewhere, then you have to imagine that that has to be a protein that sticks out from the ice. So if you see the blackness on the side of the particle, then you know that that was the side where the shadowy source was. And so the ridges for the tight junction are also, it seems like they're dark on what I think is the left side. Am I right in that? I think I am. So this is a structure that sticks up. Does that make sense? It takes a lot of time to look at these pictures.
I think so. Yeah, I'm looking at the top. There's like, what appears to be ridges or structures that come up, and it looks like the dark side is.
Okay, if we're looking here, I'll look